go ahead and get started. I know it's a nice sunny but chilly day today, so give everybody a chance to get in. A couple quick announcements as usual. First, RNA structure and catalysis, looking for courses for next term, and you've already signed up for virology and mutant viruses from hell. This is another possibility in terms of fun classes to take. I know here it also says you have to have had biochemistry, but I know you can talk to the instructor and see about checking in on that. So that's that. Second thing, a couple people have asked me about the lecture from Friday. You covered nothing, right? So I have to do everything again? Sorry. Yeah. I will try and post the PowerPoint presentation that Dr. Bartlett used. If anyone has a recording of that, please send it to me and I can post it. Yeah. Okay. Okay, great. So there's a discussion posting that has a link to the file. I will try and put that on YouTube if it's okay with you, just so it's available there too. Okay. What was I up to? This was the lecture that I gave on Thursday afternoon at the University of Florida. Yeah, sold out show, sort of, a few empty seats. No one wanted to get too close to me. It was a great time visiting with the graduate students, etc. One thing that I noticed that was very different about the University of Florida, other than the fact it was about 50 degrees warmer, was this sign, and something that we don't have to worry about too much here. So I guess we should think ourselves lucky that we're at this campus rather than their campus. So do we have any burning questions about the last couple of lectures? If we don't, I'm going to ask you clicker questions about them. So you might want to get your clickers out. Because somehow I managed to delete the clicker questions from my previous presentation, the one, I guess, last Wednesday. And then also, hopefully, we need to review some things that Dr. Bartlett talked about. So everyone got them out? Good. So way back when, we all remember last Wednesday. That was like forever ago, right? So which of the following types of transposable elements are the most common in the human genome? DNA-only transposons, IS elements, cut-and-paste transposons, retrovirus-like retrotransposons, or non-retroviral-like retrotransposons? Ten, 5, okay, we seem to be kind of divided on this one. Evidently, I didn't do a good job of telling you this, so you didn't remember, as the case may be. So pretty split across the board. It's definitely those retrotransposons. Why? Why retrotransposons in particular? Yes, we're celebrating that. Yeah. So you guessed right, as the case may be. Why particularly retrotransposons? Because they replicate. They're going to be making more of themselves each time they jump. So you're going to accumulate them in genomes, as opposed to the cut and paste and the DNA-only transposons. Nobody liked my IS elements. That's actually, in the organism, my favorite organism, IS elements are, in fact, the most common of these transposable elements. So that gets us to retrovirus-like and non-retroviral-like. What are the non-retroviral-like? What are those names for those? Lines and signs. What makes up a little over 30% of our genome? Lines and signs. So E is the correct answer. Yes? Would you like me to review lines and signs really quickly and basically what those abbreviations stand for? So lines are the long interspersed nuclear elements and the S just makes small interspersed nuclear elements. And so those are these sequences, many, many copies of them in our genome. The lines have the reverse transcriptase endonuclease gene, which is really important for cutting and then doing the reverse transcription, et cetera, to allow these to move replicatively. And the signs basically have all of the information that a line has, but it doesn't have that gene encoding the endonuclease reverse transcriptase. So it basically piggybacks on the expression of that protein in order to get the signs to move around. That make more sense? OK. Other questions on that lecture? They're not coming to you immediately now. Feel free to ask me questions again on D2L. Actually, not on D2L. Ask me an email, and then I'll respond to them via D2L. OK, so now everybody remembers everything from Dr. Bartlett's lecture, right? Good. So you can answer a clicker question on that, too. Which of the following bind to specific DNA sequences? TF2B, TF2D, TF2H, or Stedman's favorites, A and B, or A, B, and C? And don't tell me he didn't talk to you about these things. Ten, 5, 4. Again, we seem to have a bit of a difference of opinion on this one. I will, as I mentioned right at the beginning of class today, try and post his PowerPoint. Also, the recording, audio recording, again, is already on the discussions. Posted as a link there. I'll try and put it on YouTube as well. There's also last year's YouTube lecture, which I gave, which is going to be pretty similar to what Dr. Bartlett talked about. So TF2B binds to what? The BRE. Yes, it's alphabet soup time. What's the BRE? The TF2B recognition element. If Dr. Bartlett didn't mention this before, elements are going to be DNA sequences. So we'll talk a lot about elements as we move through the rest of the course here. So the response element, BRE, TF2B binds to it. That's a specific sequence in part of the promoter. OK, so definitely A. What about B? TF2D. And the TFs, these are all transcription factor for RNA polymerase II. That's what the TF2 is all about. TF2D, what's part of TF2D? TBP, the TATA binding protein. So the TATA box or the TATA. So definitely, I would say A and B. What about C, TF2H? How do you remember what TF2H does? Helicase. Does it bind to DNA specifically? I see heads shaking. I also agree. It does not. It associates with the pre-initiation complex. So that leaves us with D. Is it Monday morning? What's the deal? They're not caffeinated enough, I guess. OK, so now, assuming that he got through that material, which I assume he did, but he didn't respond to my emails over the weekend, go figure, that we now know we have messenger RNA, but that messenger RNA, unless you're an advanced organism like a bacterium or archaeum, that doesn't get translated immediately. There are things that have to happen to it before it can actually be translated. So in these prokaryotes, here, bacteria and archaea, by definition, they do not have nuclei. So the messenger RNA, once it's been transcribed by the RNA polymerase, can be translated directly. This is a very efficient way to make proteins. But it doesn't allow you the regulation, which seems to be important for us and the other eukaryotes. So that's one thing. Now, there are, as always, biology, there are exceptions. Oh, which is a reminder. Whose birthday is it today? No, it's not my birthday today. Somebody else, however, with a gray beard? Charles Darwin. Happy Darwin Day. So everything in biology only makes sense in lieu of evolution. So Theodor Dobzhansky. So evolution probably also is partly what's, well, helped or been allowed by a lot of these things that happen in messenger RNA processing. So we've talked in eukaryotes about a number of things, but one of the major ones here is that DNA and transcription take place in the nucleus, but translation, vast majority of it, some might actually take place in the nucleus, takes place in the cytoplasm. So somehow you have to go from this transcription out into the cytoplasm to get translation. There are a couple of things that happen there. We've already talked about introns and exons. So these introns are pieces of DNA which gets transcribed into RNA. So they're transcribed, but then they're removed through the process of splicing. And we'll talk quite a bit about splicing today. You also have capping, which is what happens at the 5' end. You'll notice I didn't bring my DNA model today. Don't worry, it'll be back later in the term. So you need a very specific structure at the 5' end of your RNA. And it turns out that's really important for translation, as we'll talk about later, but also for getting that messenger RNA outside of the nucleus into the cytoplasm to be translated. Another thing that hopefully is not new to any of you, the 3' end, you have a poly-A tail in the vast majority of eukaryotic messenger RNAs. How that gets put on there is another mechanism that we'll talk about. Also seems to be important for translation. Also seems to be important for getting that messenger RNA out of the nucleus. Talk a little bit about how that actual process happens, the RNA export process, and then we may or may not get around to talking about the processing of ribosomal RNA today. Potentially that will happen on Wednesday. So how does all of this happen? Usually in classes, textbooks, etc., you always talk about, okay, first you have transcription, then you have RNA processing. Turns out all this stuff is going on at the same time. And so what I like to think about, and I like this, although maybe getting a little overboard in our textbook, talking about a messenger RNA factory. And so it's not just transcription, but it's all of those messenger RNA modification proteins that are also involved in making a messenger RNA all at the same time, or at least very close one after the other. So you haven't made a messenger RNA. So maybe back up here. So look, okay, we've got this gets transcribed, then you have this primary transcript, that then gets capped and spliced and tailed, etc. No, it's all happening as that messenger RNA is being made. How is this coordinated? It mostly seems to be coordinated through the activity of enzymes which are attached to, in a non-covalent way, the RNA polymerase itself. So hopefully you talked about the so-called CTD, or C-terminal domain of RNA polymerase II. This is a long stretch of amino acids, just repeated sequences, which contain large numbers of serines. Serines are great because they've got an OH on their side chain. What are great things to do to OHs? They can get phosphorylated. And that seems to be one of the other activities of TF2H. So it's not just helicase, but also phosphorylation. So TF2H contains a kinase subunit as part of the TF2H machinery. That, excuse me, phosphorylation is important for escaping from the promoter. So once you have your pre-initiation complex, which is bound, then you have the helicase activity, then you have abortive initiation, and then finally you start to elongate. That's due to phosphorylation. But also, and important for this lecture, is that the phosphorylation of this CTD is important for interaction with other factors. Now, what's a factor again? Factors are almost always proteins or combinations of proteins. So here we have the capping factors that are associated with this C-terminal domain. As transcription takes place, there's change in the phosphorylation on this CTD, which then allows binding of proteins that are important for splicing, and finally those that are important for 3' end processing. Other name for 3' end processing is what? Poly-A tail formation, exactly. So all of this is together in one pretty ginormous complex. We'll look at some of the high-resolution images of this a little bit later on in today's talk. But really, all together, all happening pretty much simultaneously. So as transcription is taking place, the first 5' nucleotide will get modified very rapidly after it's being produced by the RNA polymerase. So these messenger RNAs, in eukaryotes, they're going to have a cap, exons, and a tail. You've got to go from intron-containing to just exon-containing and get you your tail. That's what we have down here at the bottom. Eukaryotic messenger RNAs have this 5' cap structure. We'll look at this in more detail in just a second. The coding sequence, which is just exons, and by definition, the coding sequence is going to be your just exons, and then a poly-A tail. There are also these light blue regions right here. We'll talk about these later. These are known as the untranslated regions. You have a 5' untranslated region, usually abbreviated as UTR, and a 3' UTR down here at the end. Very important for regulatory processes. Again, we'll get back to a little bit later. Another difference between the bacteria and eukarya, you notice I'm avoiding archaea here because we know very little about archaeal messenger RNAs, is that in many bacteria, you have what are called polycistronic messenger RNAs. So what the heck is a polycistronic messenger RNA? Polycistronic just means it encodes multiple completely different proteins. And I emphasize completely different proteins because a lot of the proteins that are made down here often will have differential splicing, et cetera. But at least in differential splicing, you'll have at least partly the same protein that you end up with. On the other hand, here, these are completely different proteins, usually made on one messenger RNA. That messenger RNA has a triphosphate at the 5' end, and it's usually being translated as it's being transcribed. And that's because bacteria and archaea don't have a nucleus. The ribosome and the RNA polymerases are in the same compartment, acting at the same time. What about these eukaryotic messenger RNAs? First thing that happens is, well, transcribe, but after transcription, then you have a cap. This cap is a really fascinating structure. Basically serves, again, to protect the 5' end of your messenger RNA, to help it be translated, and to help get it outside of the nucleus. So how do you do that? Okay, I probably should have brought my model today. You remember all the hungry exonucleases that are floating around inside the cell? Endonucleases, very few that cut in the middle. Exonucleases, a whole bunch that love to chew in at ends. 5' ends, 3' ends, et cetera. And we'll talk more about these exonucleases as we move along here. So exonucleases love phosphates and OHs, so you might want to block that phosphate and OH. You want to protect the 5' end of your messenger RNA. How is that done? You have this really bizarre sort of flipped over guanosine residue that gets bound to the phosphates at the end of your messenger RNA. And this is also methylated in the base itself. So this 7-methylguanosine attached to the 5' end, and in many cases you will also have methylation to the first or second of the riboses that are present in this messenger RNA. These are the cap 0, cap 1, and cap 2. All this means is cap 0 means you have no extra phosphates. Cap 1 means you have, no extra methyl groups. Cap 1 is 1 extra methyl group. Cap 2 are 2 extra methyl groups. It's mostly, again, seems to be important for stabilizing the 5' end and potentially also happening with translation. This happens after about 25 nucleotides have been transcribed. Why 25 nucleotides? It seems that that's the distance that the RNase needs to get out of the RNA polymerase itself. So to get to where those 5' end modifying proteins are hanging onto the CTD, that's about 25 nucleotides away from the active site, right in the middle of the RNA polymerase itself. So what happens? What are those factors that are important for 5' end processing? First thing is a phosphatase. ACE, what does it mean? Enzyme, what does it work on? Phosphates. We've talked about phosphatases before. Here, however, this is a nucleotide phosphatase that takes off this first phosphate, or the gamma phosphate that's present on the 5' end. That then is acted upon by one of my favorite names here, the guanylyl transferase. Took me about 3 years to figure out how to pronounce that properly. Whereby you get a GTP, which through classic mechanisms that we've heard about multiple times now, loses pyrophosphate, then a pyrophosphatase will cleave that, and this GTP now hooks up to the end right here. So we lost this last phosphate, the gamma phosphate right here, to the phosphatase, and then GTP becomes GMP and is hooked up here. Now we've got guanosine with 1 phosphate from the guanosine, 2 phosphates from that original nucleotide, and then methyl groups get added to the base, this guanosine, which gets put on the end. That happens in every single cap. Some caps then will also have riboses that are modified by methylation. So now we have at the bottom a happily capped messenger RNA that now hopefully be able to be exported, translated, and protected, et cetera. Questions on capping? The back there, yes? What does CBC stand for? CBC is the cap binding complex, which is what we will need in terms of getting that modified now cap messenger RNA out, transported, and then eventually out to be translated. There's a question down here, too. Yeah?
The second methylation on the riboses
Yep.
themselves is only for stability, or does it have any other purpose?
OK, so the question down here, I'm just going to repeat it so people in the back can hear. So what are those riboses that are methylated for? Why do they get methylated? Why do some only have 1, some have 2, some have none? The answer is we really don't know. They seem to be stabilized in some cases. Lots of viruses have differential modifications. Those methyl groups may be stabilizing the messenger RNA, but that's still kind of up in the air. I just wanted to mention it because they do exist, and they're in multiple different forms. Yeah?
So the cap 0, 1, and 2 refers to if it's unmethylated on the ribose?
Correct. So the cap 0 has no methyl groups on the riboses. We'll always have a methyl group on the guanosine. And then cap 1 will have 1 of them, and cap 2 will have 2. I don't know of any, and I don't think there are more than 2 where you have these methylated riboses. OK, so we're all happy? Good. This is the numbering here. By the way, 11 and 12, that was just relative to each of those different lectures. So here, the first one from today's lecture. How many different enzymatic activities are required for the formation of a cap on eukaryotic messenger RNA? Do not include the RNA polymerase or its specific modifications. And again, Stedman loves these numerical questions because it's really easy to come up with A through E. 1, 2, 3, 5, or more than 5. 10, 5, okay, what are the three? Phosphatase, guanyl transferase, and methyltransferase. Exactly. You notice I didn't have 4 for the methylation of the riboses because you don't absolutely need to do that. You can have cap 0 that works, so you don't have to methylate riboses. So we're not quite at 100% yet. One of these days we'll get 100%. Yeah? So the question is the methylation of the riboses done by a methyltransferase? I think more of your question is done by the same methyltransferase. And so what happens there is they're 2 different methyltransferases because they're going to be binding to very different substrates. So binding to a ribose is very different than binding to a guanosine. Yeah?
The guanyl transferase has pyrophosphatase activity itself. It's not 2 different enzymes.
Okay, so the question here is, does the guanyl transferase have its own pyrophosphatase activity? No, that's a separate, it's the same pyrophosphatase actually that's used for replication and used for transcription. And it's not actually absolutely required in this case. The other thing that I kind of forgot to mention here, if you think about 5' and 3', the way that these are hooked up, it's actually a rather strange kind of linkage because it's now a 3' phosphate on that guanine, which is on the end, attached to the 5' phosphates that are present at the very end. So you flipped over the way that these are normally functioning. So it's a 3' phosphate that came from that guanosine, which is now attached to the 5' phosphates at the very 5' end of your messenger RNA. Capping, all clear on capping? Yes? Be able to answer lots of good questions on capping on the exam? Good. So before we talk about splicing, I did want to review a particular technique. This is stuff which is not in the textbook, but should be relatively easy to find if you Google them somewhere. Something called the R-loop technique, which was absolutely critical for discovering splicing in the first place. What the heck is an R-loop? An R-loop is an abbreviation for RNA loop that forms in DNA, and it's something which can be observed with difficulty in the electron microscope. So how do you do this? You have a piece of DNA up here at the top, and you separate those 2 strands of DNA, usually just by heating them up, so denaturing or melting that piece of DNA. Again, this is a physical process. This has nothing to do with the enzymes at all. And then you mix that together with a messenger RNA, and you let the messenger RNA come together with the DNA. And in this case, you have a match between this messenger RNA and this piece of DNA. That match here will give you a double-stranded nucleotide sequence, 1 DNA, 1 RNA, and then a displaced or looped part of DNA. So it's RNA binding, which gives you a DNA loop. And an example of this is just shown over here. This is a real electron micrograph, DNA with a loop on it due to RNA. And it's a lot easier to see in the diagram down here. The 3' end here of this RNA, this is a eukaryotic RNA, that's where your poly-A tail is. And there's a short 5' extension here. If there's a 5' piece here, that means that the 5' end of this RNA doesn't actually match this DNA. Because if it's matched the DNA, it should be completely bound. You should never see extra pieces of RNA that are hanging out there. And if it's going to bind to a different piece of DNA, that starts to be really, really strange. And that's exactly what was found by, in fact, 2 different groups of researchers, Phil Sharp at MIT and Rich Roberts at the Cold Spring Harbor Lab, where they had this amazing electron micrograph, and I could show you the original, but it's really hard to interpret if you don't color code it. But it's this R loop which was done. And the R loop is basically shown here, where you have a messenger RNA bound to a piece of DNA. But the really bizarre thing here is that instead of having one looped out structure, there were multiple looped out structures of DNA which were all right next to each other, which is really bizarre, and no one quite understood what was going on. What that meant was the RNA that they had purified, again, messenger RNA, all this stuff is done in the absence of any extra proteins at all. You pull the DNA apart, you put it together with the RNA, and you just look and see what you get. In this case, it was a nice viral messenger RNA. Why viral? Because you can get a whole bunch of this messenger RNA looking at the viral genome. And what they saw in that R loop structure were these very strange loops. You know, this A loop, this B loop, and this C loop right here, relative to the DNA, which is shown down here at the bottom. So the DNA, again, part of a virus, had this light blue part that didn't bind to the RNA, but then had these little black parts that bound to the RNA, and then the green part down here at the end, on these other pieces that were completely looped out. And this was very bizarre, and took them a long time to figure out. And of course, then both of them get the Nobel Prize because it was splicing, and that's the process was originally discovered. First, people thought, oh, this just happens in crazy viruses, and soon later, people found it happened in almost all eukaryotic cells. So what this does is, the process now is modifying the RNA to make it different, missing pieces, relative to the DNA sequence. So all that is keeping exons and getting rid of introns. Exons, people found, had about 145 nucleotides. Again, on average, that should sound very familiar. Why? It's about the amount that loops around a nucleosome. So that's an interesting prospect. Also, 145 nucleotides is about a 50 amino acid piece of a protein. Most domains are about 50 amino acids. Probably also not coincidental. Introns, however, are extremely variable in size. You can have them be a couple of nucleotides in length or tens of thousands of nucleotides in length. So highly variable, and that's what's shown up here at the top. Two different human genes. The human beta globin gene, you can probably barely see the orange between all the red bits here. The oranges are the introns, whereas in some other genes, it's almost all, in fact, the majority of genes, we have mostly introns and a bunch of small exons which are in there. So this brings me to the terminology we'll be using for the next few slides. A pre-mRNA or a primary transcript, I mentioned that before. That's going to be the RNA which would be made if you didn't have any splicing going on. Of course, in splicing and transcription are happening simultaneously, you never really see these. But that pre-messenger RNA or the primary transcript has all of the introns in it. It then gets chopped out. What do you need to do this? You need 5 RNAs, lots and lots of proteins, and it's a more or less isoenergetic process. And why do I say isoenergetic? Because any bond that you break, you're also remaking. So phosphodiester bonds are broken, phosphodiester bonds are reformed. And so that process in and of itself doesn't require ATP hydrolysis because you're just exchanging bonds for each other. Those bond energies really don't change. On the other hand, as we'll see, you need a whole bunch of ATP hydrolysis to get splicing to work properly. And the key there is working properly, having specific splicing take place, because it should be hopefully really obvious that if you don't do splicing in the right place, you end up with a god-awful mess in terms of the final RNA that you end up with. So the specificity seems to be very important there, and hopefully, giving a little bit of a preview here, I'll remember what takes the most ATP in replication? The helicase. Remember that. And then, it being Darwin's birthday, evolution, probably the reason that we have these, and again, reason you shouldn't be saying this in biology, maybe the reason that we have them now, all these introns and exons probably has to do with evolution. If each of those exons corresponds to an individual domain, being able to mix and match domains allows you to have lots of variation at an evolutionary level. And we'll talk about alternative splicing. Turns out that lots of cells do alternative splicing, and probably alternative splicing is also important for evolutionary modifications that take place. So let's look at how this happens. This is the case of regular splicing. We'll talk about some bizarre splicing right towards the end of today's lecture, the beginning of next lecture. So you have now, unfortunately, they keep changing colors on us here, blue exon sequences and yellow intron sequences. So your 5' exon is going to have a 3' OH at the end, and then a 5' phosphate that's part of your intron, 3' exon, 5' phosphate, etc. That will be the 3' end of your intron. The first thing that happens is you have this very strange 2', 5' phosphodiester bond that happens between an adenine, and this is always a conserved adenine. The ribose of that adenine forms a 2', 5' bond with the 5' end of your intron. That forms what people call the lariat structure, releases the 3' OH from the first exon, which now can make a phosphodiester bond with the 5' phosphate of the next exon. This lariat is removed, and you have splicing that has taken place. So that's the chemical things that happen. How that happens is a much more complicated process. So let's take a look at it, how this happens. I mentioned the 5 RNAs. These are known as the small nuclear RNAs. I think last time or a few times ago we were talking about non-coding RNAs. These are the first of those non-coding RNAs that we're going to talk about here. So the small nuclear RNAs are U1, 2, 4, 5, and 6. 3 is important for something else. We'll get to it. It wasn't skipped. So U1, U2, U4, 5, and 6, these are the RNAs which are important for splicing. Why RNAs are so important for splicing? Probably because the way that you get specificity of splicing is through base pairing interactions. So specificity is due to those different base pairing interactions, but also the activity for splicing is catalyzed through these RNAs. So it's really an RNA-only event, although now it's developed into lots of proteins as well. So we'll talk about what these small nuclear RNAs do in just a second, but in order to talk about them, we need to talk about the sequence of exons and introns, and particularly exon-intron junctions. And so that's what we have right here. Here's our blue exon, yellow intron. The most important sequences, I've put these red boxes around here, is a GU at the 5' end of your intron and an AG at the 3' end of the intron. And as I mentioned before, there's an adenine which is going to be right in the middle of this. It's actually not really right in the middle. It's usually much closer to the 3' end of your intron. So these are the only sequences that are absolutely required for getting splicing to take place. Now, what should you immediately be thinking about when you say there's just 4 nucleotides that you need to get specific splicing? Means you're going to have these sequences all over your genome. So it means it's rather challenging to figure out, just by looking at the sequence, how splicing takes place and which of these are actually being used for splicing and which are not being used for splicing. So that process, again, just looking at sequence, you basically can't figure out what it is. What people usually do is they sequence RNAs and then go back and compare it to the DNA sequence, and that's how they figure out where splicing has actually taken place. So what about the activities of these RNAs and then a couple of proteins? So now we've zoomed out a little bit. Here's our primary transcript up here, or pre-messenger RNA. Again, here's our intron. intron is made, is transcribed. The first things that interact with this intron turn out to be proteins. They're not RNAs. There's the, again, creatively named BBP, the branch binding protein. Why the branch? That's where you have this funky 2' 5' bond, which is going to form at this adenine residue. The other protein which is involved here is the U2AF. What the heck does AF stand for? What was F likely to stand for? Factor, exactly. And here, A is activating. So the U2 activating factor, that's going to help U2, and U2 is the small nuclear RNA, to bind to this branch point. And it turns out that U2 also usually interacts here, the 3' end of your intron, which is why this branch point is usually very close to the 3' end of your intron. Now we also have U1. You now know about where the 3' end is and where your branch point is. We're going to have to take this 5' end and hook it up to here. How do you know where the 5' end is? That's where the U1 SNRN, wait a minute, what's that P at the end there? Not RNA, but RNP. Ribonucleoprotein. So there's proteins associated with these RNAs as well. So you see SNRNPs here. So these are the ribonucleoproteins. U1, U2, U4, U5, U6. So you've had U2, U1, and then U4, 5, and 6 come into this complex. So U1 at the 5' end, U2 at the branch point, and 3' end. 4, 5, and 6 will bind to this structure, and then, mostly through the activity of U6, which is why it's in red here, particularly important, then can catalyze this first reaction, which is, again, the 2', 5', and then this attack between the 3' end of the upstream exon and the 5' end of the downstream exon. In order to have all of this RNA-RNA interaction, you need to have RNA-RNA binding, but that RNA-RNA binding is going to change. How can you get RNA-RNA binding to change? You've got to get those double strands apart from each other, which needs helicases. And these are now RNA helicases. So they're going to be pulling apart strands and putting them back together. And that's where you need most of the ATP for splicing to go on. Zoom in a little further now. Let's look at the U1 interaction at the 5' end of the intron. Again, these GU base pairs here are going to base pair with an AC present in this U1 RNA. Undergoes rearrangement, again, because of the RNA helicase activity. Now the U1 is no longer bound. Now U6 is bound here. The branch binding protein, rearrangement here. This rearrangement happens because U2 is bound to other things. Now it can bind to the branch point. And specifically here what happens, this A, where you're going to get the 2' OH bound to, is squished out of the rest of the RNA so that that 2' OH is available to make the covalent bond to the 5' phosphate. And then the most convoluted gymnastics happens here. Now U6 we already have bound to the 5' end. That's what we saw that happened up here. And that's associated now also through base pairing interactions with U2, which is how the 5' end gets brought close to this branch point. And then lots and lots of RNA helicase activity happen. U5 is brought into the complex. Interactions between U6 and U2 are changed so that you can have the interaction between the 5' end, let's say the 5' exon, the 3' end thereof, and then the 5' end of this next exon. So that's the process. It's now known in exquisite molecular detail exactly how this happens. I can't follow it. It seems that every week there's a new publication that comes out telling us more about these structures. But I did want to show you a couple of these mostly to emphasize what I just talked about. I'm not going to ask you all the details of each of these individual structures. I can't remember all the details of these individual structures, so I can't expect you to. But important here is if you look at the structure, which is down here, which has been solved through both X-ray crystallography and electron microscopy, is this U6 snRNA with these metal binding sites. That's what all of these little dots are right here. And those little dots are what's giving you the catalysis. So it's the RNA which is undergoing catalysis of this first formation of the lariat, but then the linkage of the two exons together. So it's that metal binding, again, through the U6 snRNA. Mentioned before that it's U4, U5, and U6 come as a complex. They only are going to bind to your splicing interactions once you already have U1 and U2 bound to an intron. They come in as this pretty huge structure here, but also it's not just the RNAs, you have the proteins. So SNU66, no, I'm not going to ask you about SNU66. One of the proteins which is associated with this whole complex. So SNU114, U5SM, all of these different things, so the RNP, ribonucleoprotein complex, come into the structure, then have all these rearrangements. Many of these proteins are RNA helicases with ATPase activity to allow all of these structures to rearrange relative to each other. And this is just a zoom in that same paper here. This is that structure. Now just looking at the RNAs, you'll notice the RNAs are relatively small compared to all of the gray proteins that are present here in the background. Before you have splicing take place, U4 and U6 are very tightly bound to each other through base pairing interactions, but those base pairing interactions have to come apart before U6 can actually have its activity. So again, you need RNA helicase activity in order to get this structure to move forward. And then the very last of these is you can see where the structures, obviously, right here. I can't figure out what's in this particular structure. Where you have this massive amount of RNA, a whole bunch of protein, all coming together to do basically one really small thing, which is that phosphodiester bond formation. So just like we talked about really briefly with protein enzymes, all this structure around the outside for a very small part, which is the enzymatically active site. Same thing is true in splicing. All kinds of proteins and RNAs, just getting you the specificity of formation of one specific phosphodiester bond. Splicing. Okay. Which of the following is thought to contain the splicing enzymatic activity of the, oops, that should be the spliceosome, not the ribosome. Where the heck did I get that from? Must have been too late at night when I wrote this particular question. So, the spliceosome. U2AF, U1, U4, U5, or U6. We're going for that 100% today, right? Spliceosome. And zome, again, is nucleotides plus proteins. Ten. 5. I see clickers flying all over the room. That's not 100%. We still need to work on this a little bit more. Yes, it's U6. I thought I'd beaten that kind of over the head, but I guess not. U2AF is just binding. It's not providing enzymatic activity. U1 is binding to the 5' end of your intron. U4 basically actually seems to be slowing down the reaction by holding on to U6. U5 we haven't really talked about, and U6 was the one that was in red. So anytime you remember which ones were in red, that will bring you down to B and E immediately. Okay, so one of the big questions that has been bothering people actually for years, one of the people I talked to, one of the professors at University of Florida is actually working on exactly this question, is how do you not get all kinds of problems that happen with splicing? And how does the cell know how to do appropriate splicing as opposed to inappropriate splicing? So if you think about a primary transcript or this pre-messenger RNA, you've got really small exons and massive numbers of introns of all kinds of variable different sizes. So there are a couple of mechanisms that seem to have evolved to make sure that only exons are used and introns are all cut out. One of those is that these exons are all pretty much the same size. So around 150 nucleotides. And oh, that's actually shown on the next slide here. The other one is that most of these exons have proteins that bind to those exons specifically. And those are the so-called SR proteins. Why are they called SR proteins? Because they've got a lot of serines and arginines in them. So that's the only reason that they're known as such. And so if you look here at a typical pre-messenger RNA, you'll have these, I guess, what are they, purple ovals and circles here in the middle, where the SR proteins are bound to a particular exon. Those then will help binding by U1 and U2 through the binding of U2AF and the branch binding protein. So the other thing, and the other reason that you usually have splicing that takes place that is more or less correct, is that splicing and transcription are happening at the same time. So it's a co-transcriptional process, which means that the first exon is much more likely to get hooked up to the second exon than the first exon to the 15th exon. Because that 15th exon hasn't even been made by the RNA polymerase yet. So there's no opportunity for that to actually happen. Another thing that I needed to mention here is probably one of the most confusing parts of splicing for anyone are these HNRNPs. What the heck is an HNRNP? Well, the N is nuclear. OK, that's good. RNP, ribonucleoprotein. OK, so RNA and protein. H, heterogeneous. Heterogeneous means it's a mixture of all kinds of nuclear ribonucleoprotein complexes. Why do they try and torture us with all of these things? We'll see that these HNRNPs is actually very important for the final export process. So if you have HNRNPs bound to introns, what does that mean? If your HNRNP is still part of this messenger RNA, you don't want to be exporting it because it means you haven't cut out these introns yet. So, and we'll talk more about those a little bit later on. So basically, what I'm talking about here is why we're getting the appropriate splicing taking place is then to avoid these kinds of things down here at the bottom. So this is just a cartoon format. Here we have a pre-messenger RNA or primary transcript with 3 exons in it. If you have incorrect, or sometimes we'll talk about alternative splicing, maybe you'll skip this exon completely, and exon 1 will be hooked up to exon 3. Or, and this is something that happens actually in quite a lot of genetic diseases, is that exons get spliced to the wrong part of another exon. And that's mostly because these sequences, you remember the consensus sequence, you only got 4 nucleotides that are actually completely conserved. And so those dinucleotides are often found in very different places. So this kind of incorrect splicing, sometimes we'll call it alternative splicing, happens quite a bit. So we mentioned this before. If you look at the distribution of sizes of exons, and this is now from 3 different eukaryotic genomes, you'll notice the peak here is right around 100 to 150. So almost all the same size, maybe because of the same size of the nucleosome, maybe because of the same size in individual domain, but those are the pieces that get put together. On the other hand, intron sizes are all over the place, and they do seem to vary quite a bit between our 3 model organisms, humans, Cinnamomyrmex elegans, and Drosophila. In the human genome, introns have a tendency to be much bigger, and as I'm sure all of you remember, the worm and fly genomes, in terms of the DNA, are considerably smaller. But in terms of the number of genes, they're very similar. So one of the big differences in terms of the sizes really seems to be the difference in intron sizes between these various different genomes. As if this process weren't complicated enough, there are what people call alternative spliceosomes. But this, I think, the reason we talk about this is because it sort of emphasizes that splicing is generally a pretty non-specific process. Where does the specificity come from for splicing? Those small nuclear RNAs, remember, those are the ones which are base pairing, which are the ones that determine where you are going to splice. U1 and U2, because they're the ones that are binding the branch point, 3' end, and 5' end of any of these given introns. So there are other RNAs, but the rest of the process is basically identical, which will bind to now different sequences at the 5' end over here. I'll get my pointer to work. The 5' end and the 3' end of your intron, and with otherwise the same U4, U5, U6, can splice different introns out. And so all this means is it's really the specificity that you're getting from those RNAs which determines which of those introns are getting cut out, and the binding of U1 and U2 is really going to, it determines whether you have splicing take place or not. And there's this really bizarre mechanism that was found recently where you can actually take 2 different RNAs, and as long as they've got the right sequences at either end and some extra small nuclear RNPs, you can have 2 different RNAs which can then get spliced together. Because there's no particular reason, once you've got a lariat that's formed, that you wouldn't be able to bring these 2 ends together. The reason they're close to each other is because of all these RNA-RNA interactions. So there are a few cases. It's pretty rare. But you can actually have what's called trans-splicing that takes place. So 2 different RNAs that then get hooked up together because of the presence of sequences that look like an intron at either end. Yeah?
Do you ever see splicing like some sort of second time on introns? Are those then spliced once more?
So I guess the question, pardon me if I'm getting it, correct me if I'm getting it wrong here. So do you ever see 2 different splicing reactions that happen on one specific intron? So cutting.
Like after a sequence, like say some 500 base pair sequence has been spliced out.
Yeah.
Would that be then spliced again by another splicing complex?
Okay, so I guess the question is, can you have multiple splicing that happens like an intron? For instance, you've cut out a lariat, and then there will be other splicing that happens on that particular intron. I do not know of any examples of that, but there's some very bizarre circular RNAs that we may have a chance to talk about later on in the course that were just discovered about 5 or 6 years ago and maybe addressed with some of these things. So hold on to that thought, and we may get back to it. Yeah?
Back a couple slides.
Yeah.
So you said that at the U1 and U2 determine where to splice.
Mm-hmm. Yeah, it's right here.
SR proteins, is that what they were in the back
Mm-hmm.
slides?
Yeah.
If there's, what is, you guys missed something. What is the purpose of those if the U1 and U2 determine where to splice? What is the purpose of those binding specifically to those exons?
Yeah, so again, I'll try to paraphrase your question here, see if I get it right, is If it's just U1 and U2, why do you need all of these SR proteins? This is, again, to sort of paraphrase your question. The reason seems to be that all that you're binding to is a dinucleotide, GU here, and AG here. You're going to see these all over the place in your genomes. If you think about two nucleotides right next to each other, 1 in 16 are going to have those two right next to each other. If you've got 6 billion base pairs in your genome, there are going to be a heck of a lot of these. And particularly if you think about these introns, some of them being literally 10,000s of nucleotides long, there are going to be lots of GUs and AGs in there. And so those specific AAUs and AAGs, that's where you need these, SR proteins, things that are defining where exons are. Okay, so we have these SR proteins, et cetera, et cetera. And I talked a little bit about the different exons and introns. Most exons in most genes get used. And that's because we have co-transcriptional splicing that happens. We have the SR proteins that are associated with individual exons. But it turns out that it's not all the exons in most genes that get used. It's just the majority of exons in most genes gets used. And this was only actually really known when we started to look at RNA sequences specifically from the human genome. If you just look at RNA sequences you find in the human genome, there are tens of thousands of different RNA sequences, messenger RNAs. Well, how can you have tens of thousands of different messenger RNA sequences when you only have 20,000 genes? So there were a number of companies that were selling the hundreds of thousands of genes that were supposed to be in the human genome. And when they finally finished the human genome, they found, uh-oh, there are a lot fewer of these. Well, what's going on? Well, it turns out that there are modifications, mostly in terms of splicing and how you get these individual pieces. And actually, between 60, and people argue actually about how often this happens, at least 60%, I should probably say, of human messenger RNAs are going to be alternatively spliced, whereby a few exons are going to be slightly different. And that's shown here for one particular gene, the alpha-tropomyosin gene. Here we've actually got relatively large, and I don't like this particular image here because the introns should actually be bigger, but all of the dark brown are the exons, and then the light brown are all of the introns. And in different tissue types, different kinds of muscle, fibroblasts, et cetera, you have different exons that are actually being used. And as far as I know, the record holder for this is a particular gene in Drosophila, not that important what the name is, but seems to be really important for neural development. It's one gene which can be spliced in over 38,000 different ways. So if you take the 20,000 genes that we had in our genome, multiply them by 40,000, get a whole bunch more possibilities in terms of how splicing could take place. How do we know that it's got 38,016 different possibilities? That's because there are these alternative exons here, 12 times 14 times 33 times 2. Most of the exons here are being used. So all the gray ones, those are present. We'll call constitutive exons in that particular protein. But then these alternative exons, which can also be used. So before I talk about how we get this alternative splicing to take place, I think we'll stop here. And again, I'll try and post the lecture, PowerPoint, et cetera, from the lecture on Friday on both D2L and then the audio on YouTube.